Hello everyone. Today we are going to start a new chapter. It is circulatory physiology, and the first topic is blood, and we will see what are the functions and what is the composition. So generally in this classification, you can see that the blood can be basically made up of two components, the plasma and the formed elements. And the plasma, it has a combination of different things like amino acids, nutrients, proteins, gases, electrolytes, even the waste, nitrogenous waste it carries. And within the protein, again, they are further classified as albumins, globulins, fibrinogens, and also lipoproteins. And within the formed elements, that are the platelets, the leukocytes or WBC, and erythrocytes or RBC. And again, within the WBC, there are two classifications: granulocytes, which possess granule-like substances in the cytoplasm, and agranulocytes without any granules. So agranulocytes are further divided into lymphocytes and monocytes, and these are the thing which is directly involved in immune response. And in the granulocytes are further divided into neutrophils, eosinophils, basophils. Most of them are produced during their abnormal number indicates certain diseases. And this is a blood smear you must have prepared during your +2, and you can see that a very clear kidney-shaped structure known as monocyte in the granulocyte. Next is round-like structure, nucleus, which is known as lymphocyte, with very little cytoplasm. Then bilobed structure, telephone-like structure called eosinophil. Then coming to multilobed structure, with though granules are present, it is not very prominent, that are neutrophils. Then also the deeply stained big granules which even cover the nucleus, they are known as basophils. And of course the red ones are the erythrocyte, and the smallest, the minute purplish color which you see are the thrombocytes. So now we will see in detail about all these things. So first is blood. So blood is generally called as a fluid connective tissue as it has a matrix. So connective tissue means, just like in the slide you can see that tissues, we know that it is made up of number of cells, and blood contain different types of RBC, WBC, and even proteins also. In the microscopic image, high resolution image, you can see that different strands of proteins are also present, and it generally take up a form of a tissue only. And it usually circulates through a network of closed circulatory system. And even open circulatory systems are also there in the very primitive or and in invertebrates, not all, but still in some invertebrates, open circulatory system is there, and closed circulatory system is mainly the characteristics of vertebrates and also certain higher level invertebrates. And generally the blood of the lower animals is watery in nature, while our blood, we know that it has certain viscosity, especially viscosity in the sense that it has some sort of, it won't easily flow. അതായത് ഇപ്പോൾ നമ്മളൊരു മിൽക്കിലൊക്കെ വിസ്കോസിറ്റി ഉണ്ട്, അതേസമയം വെള്ളം എന്ന് പറഞ്ഞാൽ ഒട്ടും വിസ്കസ് ആയിട്ടുള്ള ഫ്ലൂയിഡ് അല്ല. അപ്പോൾ അതിൻ്റെ ഒരു ഇത്, സോ ഇറ്റ് ഹാസ് എ സ്പെസിഫിക് ഗ്രാവിറ്റി which is about 1.05 to 1.06, and blood is also slightly alkaline in pH, that is about 7.4. സ്ലൈറ്റ്ലി ആൽക്കലൈൻ എന്ന് പറയുമ്പോഴത്തേക്ക് നമുക്കറിയാം ന്യൂട്രൽ വാട്ടർ പി എച്ച് ഇസ് 7. Anything that is less than 7 is acidic, and anything that is greater than 7 is basic or alkaline. So next slide we will see the functions of blood, and it could be classified into 3 categories: transport, regulation, and protection. So we know that transport is the blood is through blood only whatever waste products are generated or nutrients are there, it gets transported to body and different tissues. So here first function is transport of oxygen from the lungs of the tissues and carries back carbon dioxide from tissues to lungs for elimination. That all you clearly know, but it's like some people might be knowing that it is the function of blood. Next is it helps to transport the absorbed nutrients to various tissue for utilization. That we already covered in nutritional physiology, how blood is acting as a transport media. Then in the excretory physiology, when we learn, we will understand that it can also transport many waste products of metabolism from tissues to kidney for excretion. Then the next function, as an endocrine glands are directly released into blood. We know that the hormones, and from that the blood will carry to the target organs, and then the hormonal action will happen, and whatever products are there, that is synthesized or secreted by the cells. Then finally the meaning is same, that it can transport many metabolites to and fro between the tissue. So it's the first classification, transport. Next is regulation. Regulation in the sense that it can maintain the acid-base balance or the pH, because we know that plasma has several proteins, and in the next slide we will see the composition of plasma, and you will understand how it can act as a buffering capacity. Buffer is the substance which will stabilizes the pH. In the absence of buffer, what will happen? It's like the blood pH is 7.4. So without buffer, it can even become acidic by the end products like carbon dioxide, or some certain times very alkaline due to some release of some sodium salts. So it is the buffering capacity which actually stabilizes the pH to 7.4. Otherwise, if pH is disturbed, our whole body will get disrupted, and it can even lead to death. So it is the functions of blood only. Next function is thermoregulation. Thermoregulation in the sense we can maintain homeotherms, or endothermic animal has to maintain a constant body temperature. We know what is our body temperature is 37 degrees Celsius, and there are certain groups like frogs and all, they are ectotherms. Ectotherms, their temperature is directly regulated by the environment. The environment is cold, their blood will be cold, and if there is hot, it will remain so. But in endotherms like us, we need a proper body temperature. Even we are living in Arctic or Antarctic, all humans have same body temperature. So that function is actually regulated by blood only. Here what they do is that, for example, if the purpose is to reduce the temperature, heat generated by the activity, our normal physiological activities are distributed by circulatory system, and increased blood flow happens. This will causes warming of skin and resulting in faster heat loss. While when we have to conserve our energy, especially in cold climate and all, what body do is it will decrease the blood flow, thereby conserving the heat, and that heat we can maintain used for the maintenance of body temperature. Then even we know that I already showed that slide. You remember where blood our with increasing age the water content decreases, and that decrease is the final results of aging and ultimately senescence and death. So here the maintenance of water content also very important, and that is carried out by blood only. So that is all about regulation. Next, coming to protection. Protection means we know that we already blood contain a lot of WBCs, and it can protect our body against foreign agents such as bacteria and viruses, and they are the main players of immune system. And blood is also having clotting mechanism, especially that platelets or thrombocytes. And otherwise, if this clotting mechanism is not there in the blood, then even a small cut can be life-threatening to us. So clotting is required. Otherwise, whatever 5 liter blood we have, that will just flow out of the blood, and we know that blood has many functions, and all these functions will fail if heavy blood loss happens. And it also acts as a reservoir for substances like water, electrolytes, etc. Even in the case of this mineral metabolism, we see that transferrin and ferritin thing, it can act as a reservoir, and whenever is required, it will slowly release. Next is composition of blood. So normally the total volume of blood is called as blood volume, and in an adult it is about 5.5 liters. And in the blood there are two segments we already told, plasma and formed element. Plasma mainly constitute the fluid part, while the formed elements constitute the cellular part. And usually in laboratory it is, you have to do this practical also this year. That is to find the hematocrit value. Here what does hematocrit means? The percentage of total blood volume occupied by RBC alone is known as hematocrit. So here what we do is, normally we take blood in this small capillaries and allow to settle. Definitely the heavier will settle down, and based upon the weight, the most lighter one will remains on top. So here after this, we can see that the red blood cell will get settled on the bottom portion, and it also constitute around 37 to 47% in normal blood. Then there will be a buffy white coat like substance in between, which are made up of WBC, platelets and all. And you can see on top there is a yellowish viscous thing, which is known as the plasma, which is made up of several proteins, nutrients, hormones, etc. And it will also constitute around, and even the hematocrit value differ among male and female. Here you know the symbol of male and female, I hope. Especially that cross symbol is represented in female and the arrow one by male. So normally the hematocrit values are different for male and female. Accordingly the hemoglobin also. But in the case of anemic patients, the blood cell, the total expected RBC number will be very less due to hemolysis or some other factor. And in some cases called as polycythemia, there is a very elevated hematocrit value where the RBCs will be more compared to plasma. So this is all about the general classification or composition of blood. Next we will see a small topic, that is platelets or thrombocytes. So in the structure you must have seen that it is the smallest of all the formed elements. Its size is only 2 to 3 microns. Microns is like 10 to the power minus 6, or if you see 1 centimeter, it is again divided by 1 by 1000 is the micron. So now you can just imagine what will be the size. 2 to 3 microns look like, and these are not having a proper shape and they are look like very fragile. അതായത് ഒരു ഇങ്ങനെ പൊട്ടിച്ചിതറിയ കഷ്ണം പോലെ. നെക്സ്റ്റ് സ്ലൈഡിൽ നിങ്ങൾക്ക് അതിൻ്റെ ഫോട്ടോ കാണാം. പിന്നെ similar to RBC, it doesn't possess any nucleus, but in WBC we have seen very prominent nucleus, and but similar to WBC, it has many granules like things inside. And what is the importance is that they are very important in blood clotting. We already told in functions, without if blood clotting doesn't occur, it can be life threatening. And generally it is formed by the fragmentation of large bone marrow cells. Bone marrows are the places where the formation of RBC and WBC occurs, and its concentration is less compared to RBC. It forms only 2.5 to 5 lakhs cells per millimeter cube of blood. Next, coming to composition of plasma. So plasma, we saw in the hematocrit, it is very light, it floats. So here also, because it is 90 to 92% comprises water, and the remaining ones are made up of many dissolved materials, and this is the important factor which can regulate the pH. So there are basically 4 types of protein: albumins, globulins, and fibrinogens. Albumins are the proteins which are most abundant and they are synthesized in liver. Then comes globulins. Globulins are further divided into alpha, beta, and gamma globulins, and gamma globulins are the ones which become antibodies. Because you have seen that antigen, any pathogen or antigen, when it enters our body for immune reaction, it will produces antibodies. So antibodies are actually the part of plasma proteins. Next comes fibrinogen. Fibrinogen, along with the thrombocyte, they are also important in blood clotting. Then another group of proteins are known as lipoproteins, and these lipoproteins are actually act as a carrier molecule. We saw that this ferritin and all, remember, iron is bound to one protein, we told, and in that only it will transport throughout the body. So similarly, lipoproteins are the one which are carriers of all these fats and organic compound, and their presence is required to maintain proper osmotic pressure. Otherwise, we also know the role of blood in blood pressure control and all. So these are the lipoproteins are the major ones which are involved in that. And normally when you do some biochemical test like testing of sugar, testing of proteins and all, it is the serum part which we use to take for different type of analysis. So serum is nothing but the fluid portion of blood after removing the clot, and basically it is a plasma from which the fibrogenous and other proteins involved in clotting also have been removed. അപ്പോൾ serum ആണ് ഏറ്റവും, in comparison to plasma, serum is most the lightest one. കാരണം അതിൽ ഓൾറെഡി ബ്ലഡിൻ്റെ എല്ലാം എലമെൻസും മാറ്റും, അഡീഷണലി ഈ ചില പ്രോട്ടീൻസും മാറ്റിയിട്ടുള്ള കിട്ടുന്ന ആ പാർട്ടാണ് നമ്മൾ കൂടുതലും ഈ ബ്ലഡ് ഷുഗർ ടെസ്റ്റ് ഒക്കെ ചെയ്യാൻ പോകുമ്പോൾ നമ്മൾ ബ്ലഡ് എടുത്തത് നിങ്ങൾക്ക് ഓർമ്മയുണ്ടാവും, പക്ഷേ അതിൻ്റെ സിറം പാർട്ടാണ് used for analysis. Next, coming to the organic components, though it constitutes only 0.2 to 0.3% only, and these organic components are hormones, it can be vitamins, enzymes, glucose, amino acid, nitrogenous waste, and other metabolites. So all these things are for detection, we require to separate the serum out of it. And it also has many inorganic constituents, like major portion is sodium ions, and the other things are potassium, calcium, and magnesium. So sodium exists in different forms like sodium chloride, sodium bicarbonate, and sodium phosphate. Now all these salts are required in the buffering action. Buffering action means where we have to maintain the pH. Next is formed elements. So these are the formed elements which already I discussed about this. And in the lymphocyte you can see there are two denoted by two colors, because in lymphocyte actually there are two types, B cells and T cells. Both are involved in immune response, and in immunology we will study the same in detail. And coming to erythrocytes, of the blood formed elements, 99% is contributed by WBC only. And the erythrocyte, it's maybe in your many quiz and all they ask, or in the PSC question they ask that normally erythrocytes are nucleated in majority in all mammals excluding the camels. In all vertebrate except the mammals. So mammals are having a RBC without any nucleus, while and in camels also, mammals in the sense mammals are even you must have I already studied the classifications like fishes, then comes amphibians, reptiles, apes and mammals. So in that only the mammalian erythrocytes are non-nucleated. There is no nucleus. While in all other vertebrates starting from fish to bird, the RBC contains nucleus just like WBC. But within the mammal, the only exception is camel, whose blood is nucleated. And in the structure you can see it is already circular, but it has some biconcave in nature. That is, one side is having some depression like thing, and and normally this hemoglobin is this you will be studying more in your respiratory physiology topic. For time being, you just understand that hemoglobin is an iron containing molecule with four polypeptide molecules are attached to it, and the overall structure is shown here. Next, coming to production of erythrocytes. So we know that in the mammals RBC is non-nucleated. So normally the production of hence what happens is, since there is no nucleus, it cannot live like a lifelong like RBCs or T cells and B cells or lymphocytes. So it can live maximum up to 120 days only, and this RBC will go to their graveyard, which is known as spleen, and it is estimated that every day 1% of RBC is getting destroyed, but at the same rate it is also new RBCs are getting formed. And normally this process is regulated by erythropoietin hormone, which is secreted mainly by kidney and liver cells. So production of erythrocyte image is shown here. So I told that RBCs are basically produced in bone marrow cells, where some for their growth and activity certain growth factors are also present. And RBC is produced within day 5 or 7 only, the one which is in our blood. First, in the day 1, it is started from a stem cells. Stem cells means those cells which has the potential to undergo to make into any kind of cell. So in the stem cell hemocytoplasm, in the day 1 it looks like this. Then it becomes in the second cell called proerythroblast, it becomes committed in the sense from this stage onwards stem cells are committed to become RBC, not to other muscle cells or nerve cells, but the stem cells has the potential to become anything in the body, any cells, muscle cells, nerve cells, or red blood cells or WBC, anything. But here, after the second stage known as pro-erythroblast, it becomes committed. Means it will now only it has the potency to become a erythrocyte. Then the first early erythroblast stage, which happens in day 2, ribosomes are synthesized. We know that ribosomes are the cell organelles which are involved in protein synthesis. So in the day 3 and day 3, day 4, day 3 and day 4 time, this erythroblast will start, from this ribosomes will start producing hemoglobin, because hemoglobin is a protein. Afterwards, the cell nucleus size decreases in day 4, day 5 time, and finally ejection of nucleus happens. Then it becomes reticulocytes, and finally it is released in the form of RBC or erythrocyte, and it can circulate in blood even up to 120 days, but later on it gets destroyed in the spleen. And in certain jaundice and all, the destruction rate is very fast. Suppose it is only 1% per day. During jaundice, it can go very high. And this is the normal RBC count for male and female. So females per millimeter cube, there are 4 to 5 million. Million means 1 million is like 10 lakh. So around 4 to 5 million RBCs are present per millimeter cube of the body. And also in male it is around 5 to 6 million per millimeter cube of the blood. So this is calculation you will be doing when you do the practicals, where we have to differentially count the RBCs and WBCs. Next, coming to leukocytes. So leukocytes, also known as WBC, I already discussed the structure, and here the size is also given. We'll remember micron is like 1 by 1000 of 1 by 1000 of what you have learned in this thing. So WBCs, in the WBCs, the major one is neutrophils, and it's generally it will be around 50 to 70%, while eosinophils are less than 5%, and normally that 1 to 5% is enough for a healthy individual. Basophils are the rarest of the WBCs, and they are present less than 1% of the total leukocyte. And lymphocytes which are the involved in our immune system, that is B cells and T cells, they are around 25 to 30%. And monocytes are very big compared to all other leukocytes. It circulates in our blood around 3 to 9%. So anything number less than that or more than that indicates certain type of diseases or disorder in the blood. And usually these leukocytes are stained with Giemsa stain, which is specifically stained the phosphate group of DNA, where there is adenine-thymine bonding. We know that adenine pairs with thymine, guanine pairs with cytosine, and the DNA structure, other than nucleotide, there are phosphates also. So this is the phosphate groups which are actually stained by the stain, and with the help of stain we can see clearly under the microscope. So now we will see the functions. Neutrophils are having a phagocytic activity. Phagocytosis, we already see how it is happening in the cell. Remember the membrane transport. So what they do is they phagocytes the foreign cell toxins and viruses, and inside the cell, with the help of lysosome, they will just degrade all these foreign particle, and normally it stains light violet, and since the granules are very small, it is difficult to see or differentiate these granules under the microscope. and they are very multi-lobed, connected by thin strands of nuclear material. This is all about neutrophils. Next is eosinophils. Eosinophils are also phagocytic, but they are weak phagocyte, and generally their phagocyte is remove the antigen antibody complex, because we know that when antigen or pathogen is encountered in the body, our body, especially the plasma proteins and all, will produce the antibodies, and these antibodies antigen complex later on will get eliminated from the body with the help of this eosinophils. And here the granules are generally are acidophilic in nature, and hence with this Gießma stain the granules will stain pink, and here also there are bilobed structures, and these two lobes are connected with the help of a nuclear strand. Next, coming to basophil. Basophil's granules are biggest among all the three granulocytes, and because these granules itself contain certain histamines and heparin. Histamines are the vasodilators, and vasodilators എന്ന് പറഞ്ഞാൽ ഇപ്പം നമ്മുടെ ബ്ലഡ് വെസൽസിൻ്റെയൊക്കെ പമ്പിങ്ങിന് ചിലപ്പോഴൊക്കെ ബ്ലഡ് ഡൈലേറ്റ് ചെയ്യണം, അതുപോലെ തന്നെ കൺസ്ട്രിക്റ്റും ചെയ്യാറുണ്ട്. അപ്പോൾ പ്രോപ്പറായിട്ട് ഇപ്പോൾ ഡൈലേറ്റ് ചെയ്യാൻ വേണ്ടി അല്ലെങ്കിൽ ബ്ലഡ് വെസൽസിൻ്റെ ആ സൈസ് capillaries size increase ചെയ്യാൻ വേണ്ടി vasodilator is required. So that function is contributed by basophil, and it has this anticoagulant heparin. Anticoagulant means that always if there are thrombocytes and all which can actually coagulate the blood, which is helping in not coagulation, sorry, blood clotting and all. So for smooth function means when blood has to flow through the body, if some blockage or coagulation happen, it will block. ഇപ്പം നമ്മൾ ബ്ലോക്ക് എന്നൊക്കെ കേട്ടിട്ടില്ല, അല്ലെങ്കിൽ സ്ട്രോക്കിൻ്റെയൊക്കെ സമയത്ത് എന്താ പറ്റുന്നത്, ബ്ലഡ് വെസൽസ് ബ്ലോക്കായി പോകും. അപ്പോൾ അങ്ങനെയുള്ള ബ്ലഡ് വെസൽസിനെയൊക്കെ ബ്ലോക്ക് ചെയ്യാതിരിക്കാൻ നമ്മൾ എപ്പോഴത്തേക്കും ആൻറ്റികോയാഗുലൻ്റ് കൊടുക്കും. പിന്നെ ഇൻ ലബോറട്ടറി ഇപ്പം നമ്മൾ ബ്ലഡ് ടെസ്റ്റ് ചെയ്യാൻ വേണ്ടി ബ്ലഡ് എടുത്ത് ബോഡിയിലോട്ട് വെച്ച് മീൻസ് ഔട്ട്സൈഡിലോട്ട് കൊണ്ടുവരുമ്പോൾ തന്നെ എന്ത് പറ്റും, ബ്ലഡ് കട്ട പിടിക്കും. പക്ഷെ ബ്ലഡ് അങ്ങനെ കട്ട പിടിച്ചു കഴിഞ്ഞാൽ ഈ പറഞ്ഞ പോലെ ചിലപ്പോൾ നമുക്ക് ചില ഫങ്ഷൻസിന് ടെസ്റ്റിന് അത് പറ്റൂല. അപ്പോൾ അതുകൊണ്ട് തന്നെ എന്താ ചെയ്യണമെന്ന് വെച്ചാൽ we need certain we provide additional anticoagulant, so blood will not get clotted. And here the granules appear deep blue to purple color and even mask the nucleus. And here also the nucleus is lobed. Then is lymphocyte. Lymphocyte, we told that they play an important role in immune response. And here the B cells are involved in antibody production, and T cells are mainly formed, and these cells can act against the viral infected cells and tumor cells. And here the nucleus almost fills the cell, which stains dark purple. Hence cytoplasm is very clear. Next is monocyte. Monocyte are ഏറ്റവും biggest, like around 16 to 22 micrometer in size, and since it is biggest, the moment inside the blood, monocyte leaves the blood by a process known as diapedesis. And when this monocyte once leaves the blood, it can turn into macrophages. Macrophages are also having a high phagocytic action, and it helps in the body's defense against bacteria and viruses. And here the nucleus is often kidney or bean-shaped structure, and here the cytoplasm is more compared to the lymphocyte, and it can stain light blue. Next topic is abnormalities in blood cell count. Abnormalities mean I already told that anything which is increasing means there is a proper level to be maintained, and anything which is beyond that or less than that indicates certain abnormalities. So coming to RBC when it is increased than usual number, it is known as polycythemia, and it can be normal or like just because of some sudden changes in the environment, or can indicate certain diseases. So generally, like tumors can also increase the count of this RBC. Then especially we know that RBC's main function is it acts as a respiratory pigment, hemoglobin. So when in the areas where there is not enough oxygen is there, then definitely the blood will release more and more RBC, so it can carry the respiratory pigment hemoglobin without any problem. So in all this case, polycythemia can occur, and the decrease is named as anemia. Anemia is a very common word nowadays hearing, and especially we women are anemic because they have they will lose certain blood during their monthly cycle. So here anemia is also contributed by iron deficiency, because we know that iron is the in the hemoglobin, iron is the center. Iron is actually joining the four polypeptide chains. So if iron also not there, hemoglobin will remain fragmented. And in some diseases, especially it occurring in some areas, we call as sickle cell anemia. Here RBCs are like sickle shape. Sickle cell in the shape. So what happens is it will reduce their capacity for oxygen transport. And also when very heavy blood loss occurs during accident and all, we become anemic. Next is platelets. Platelets increase is known as thrombocytosis. In the thrombocytosis, what happens if platelets is very high, then even it can occur, spontaneous clotting can occur. Spontaneous and in case of thrombocytopenia, where the platelet count is low, the clotting is very delayed. And we know that if clotting is delayed also it's a problem. Like if some cut happen and if clotting is delayed, then we will lose lot of blood. And normally the platelets count are abnormally decreased during dengue fever. Even they need a platelets transfusion, just like blood transfusion. During dengue, people has to take additional platelets inject in their body. Then is neutrophils. So neutrophils abnormally it will increases, especially during certain conditions like appendicitis, smallpox, chicken pox type smallpox and then rheumatic fever, that is vadapani. While it decrease is mainly occurs when there is a viral infections like influenza, rubella and hepatitis. This time like neutrophils count generally its concentration is 50 to 70%. Next is eosinophil. Eosinophil or eosinophilia, it's a very common term, and even those who are living in the coastal areas also will have a slightly higher eosinophil count. The abnormal or increase in eosinophil cells denotes any parasitic diseases, and also those who are allergic, like dust allergic, normally they have a high eosinophil count, and then asthma case eosinophil will be higher, and the reduced number of eosinophils, like if it is go below 1%, that condition is known as eosinopenia. And it usually occurs when our body is really stressed. Next is basophils. Basophils are like basophils, I already told it is already less than 1%. So if it is sudden increase happen when there is a chronic infection, When there is a chronic infection, it indicates certain hemolytic anemia or chicken poxes. Next is lymphocytes. Lymphocyte, we know that lymphocytes are the one which produces B cells and T cells and involved in immune response. So definitely any infection occurs, the number of B cells and T cells will be higher. And when it is decreased is that one classic example is AIDS, where our immune system is attacked. And also some similar other immunodeficiency diseases, also lymphocyte count will remain less. For example, we know that COVID-19 and all, we are highly susceptible, and we are not, our body sometimes fail. For some people, it fails to produce enough antibodies. So because it actually hampers the production of lymphocytes. The last one is monocytes. Monocytes increased number is known as monocytosis, and their high number is observed during malaria, then Rocky Mountain tick-borne fever, Rocky Mountain spotted fever, and also during endocarditis. So this slide is the last slide I just kept, because we know that blood has a lot of functions, and it is our duty to preserve the blood and to boost our immune system, and this we can do by simply eating selective foods. For example, these are the examples for iron-rich food. And I already told that iron absorption is dependent by vitamin C concentration also. So to increase the iron absorption, we have to also take vitamin C in enough quantities, and which is mainly comprised of citrus food and colored food. And these are the platelets. For example, in dengue and all, these are the top food to increase the blood platelet count. Even papaya leaf, some people are using during this dengue fever to increase the platelets count. And these are the white blood cells immune foods like strawberries and all. And finally, our whatever problem in the blood is reflected in our skin also, രക്തദോഷം എന്നൊക്കെ പറയും. So if you want a very clear skin, your blood should be highly, we have to purify our blood, and we can purify our blood by adding certain things in our diet, like turmeric, we already do then, and alone green tea, which has antioxidant capacity, lemon and all also will purify our blood and glow our skin. So eat healthy and take care of your blood. Thank you.